Welcome to the deep dive. We take your sources, research notes, whatever you've got, and distill the key insights. Think of it as your shortcut to being well informed, with hopefully a few surprises along the way.
And today, we're diving into something pretty fundamental. Human evolution, our genes, and this really deep link between our biology and, well, our culture.
Yeah, kicking off with a mind-blowing fact. Humans share over 98% of their DNA with chimpanzees. Okay, let's try and unpack this a bit.
It's staggering, isn't it?
It really is. So this deep dive is all about understanding that incredible journey, from a quadruped ape ancestor to, well, to us. Bipedal, big-brained humans. We'll look at everything from the tiny biological bits to the big cultural shifts.
And what's really fascinating, I think, is that it's not just biology. It's how deeply intertwined that biology is with culture. We're going to connect those dots, genetic changes, unique human behaviors helping you understand not just what happened, but maybe why it matters so much.
Right. Our mission today is basically to give you a clear, engaging tour of physical anthropology. We want to make these complex ideas feel, well, graspable, and hopefully get you thinking critically about what it means to be human, maybe spark a few aha moments.
Exactly. We'll dig into the core principles of human biology and evolution, reveal some surprising reasons behind our modern traits. Forget maybe the high school biology. Remember, this gets closer to the chemistry of life, the reactions that make us us. And trust me, the insights are definitely worth it.
Okay, so that 98% similarity with chimps, if we're so similar genetically, how did we end up so, well, different? Where do scientists even start placing us on the big tree of life?
Right. Classification is the starting point. Biologists begin with the concept of a species, basically organisms that can mate and produce fertile offspring, like all humans are Homo sapiens. We can all interbreed, and our kids can too.
Okay, so like a horse and a donkey. They can make a mule, but the mule can't reproduce, so different species.
Perfect example. Exactly. Then you go up a level to genus. That's a group of related species from a common ancestor. Lions and tigers, they're both in the genus Panthera. Humans, we're in the genus Homo.
Got it. Species, then genus.
Then family, that groups multiple genera. So lions, tigers, domestic cats, they're all in the Felidae family. And for us humans, we're part of the great ape family. That includes chimpanzees, orangutans, bonobos, our closest living relatives.
Okay, so we're right there with them. And the human story then essentially kicks off with a common ancestor, some kind of ape, maybe 7 or 8 million years ago.
That's the idea. An ancestor whose lineage split one path leading to chimps, the other eventually leading to us. That's our starting point.
Right. Now, what really sets us apart, most people jump straight to intelligence, the brain.
And they're not wrong. Our brain is remarkable. But that incredible evolutionary leap, it came at a pretty significant cost.
The cost for having a big brain, that seems counterintuitive. What kind of cost are we talking about?
Energy cost, a huge one. Consider an average mammal, say 60 kilos. Its brain is maybe 200 cubic centimeters. Our ancestors are around the same weight, maybe 600 cc. Modern humans, same weight, but 1,200 to 1,400 cc.
Wow, that's a massive jump, 6 or 7 times bigger than the average mammal.
Exactly, and double our more direct ancestors. But here's the cost. That brain, while only 2-3% of our body weight, devours about 25% of our energy, even when we're just sitting around.
25%, just thinking or existing.
Just existing. It's incredibly power hungry. This created a constant fundamental pressure to find food, and it led to a trade-off. Muscle atrophy. Our muscles became weaker.
So we sacrifice brawn for brains, essentially.
Pretty much. Humans are physically much weaker than chimpanzees. You all know Harari's line. You might win a debate with a chimp, but physically it could tear you apart. That energy went to the brain, not massive muscles.
Okay, brain power at the cost of muscle power. What about the other big human thing, walking on 2 legs? Bipedalism. What were the gains and losses there?
Well, the huge gain was freeing up our hands. Massive advantage for tool use, carrying things. But yeah, there were definitely costs. The immediate ones are things we still feel, backaches, stiff necks.
Tell me about it.
Right. Your body weight, instead of being distributed across 4 limbs, is now transmitted linearly down your spine to just 2 legs. That creates a lot of pressure.
Okay, so everyday aches and pains. But you mentioned a bigger cost, especially for women.
Yes, a really critical evolutionary dilemma involving the female pelvis. For efficient walking, the pelvis needs to be relatively narrow. If it's too wide, you waddle, it's inefficient.
But it also needs to be wide enough for childbirth, right? For the baby's head to pass through.
Exactly. A head that's getting bigger because of that brain evolution we just talked about.
Yeah.
So, narrow for walking, wide for birthing. It's a biological conflict.
So how did evolution solve that problem?
Nature's solution was pretty ingenious, if demanding.
Hmm.
Give birth to babies whose brains are still very immature. About 90% of human brain growth actually happens after birth, mostly in the first few years.
90%. Wow. So babies are born neurologically unfinished, basically.
Very unfinished. And completely dependent. They need constant care, feeding, protection for years, decades even. This immense dependency created a powerful selective pressure for strong social bonds.
Ah, so that's the connection. Because a mother trying to raise this helpless infant alone would be incredibly vulnerable.
Exactly. Early humans who formed groups who cooperated where others could help provide food and protection while mothers care for infants, they had a much higher survival rate. This biological constraint, this need to care for dependent young, is arguably a key reason we evolved into such intensely social animals.
That makes so much sense. Biology driving social structure. Okay, so this whole journey, it wasn't just biological tweaks. Discoveries made a huge difference too. Let's talk about fire. That seems massive.
Oh, fire was a complete game changer. It provided this huge, almost limitless source of energy. And crucially, it offered cultural solutions to environmental problems.
Cultural solutions. How so?
Well, first, obvious one. Light. It extended our active hours beyond daylight. Huge advantage. Second, warmth. We're warm-blooded, right? Regulating body temperature takes energy. Fire let us migrate into colder climates. Think Homo erectus heading into Ice Age Europe without needing to biologically evolve thick layers of fat, like, say, a polar bear.
Mm.
We used culture, fire, to bypass a slow biological adaptation.
A technological fix for a biological problem. Clever. And what about cooking? That must have changed us physically.
Profoundly. Cooking makes food softer. This reduced the need for massive, powerful jaws and large teeth to grind tough, raw food. Over time, our teeth got smaller, our jawbones shrank.
Is that why so many people need braces? Our jaws got smaller, but we kept the same number of teeth?
That's a big part of it. Malocclusion, crooked teeth, it's partly a consequence of having the same dental toolkit in a smaller space. Thanks, cooking.
Huh, okay. Smaller jaws. What else?
Less chewing effort. Chimps spend maybe 5 hours a day just chewing. Humans, with cooked food, maybe an hour. That saves a ton of energy. And cooked food is easier to digest.
Which means,
Our intestines could get smaller. Digestion takes energy too. A smaller gut needs less energy to run.
okay, so energy saved from chewing, energy saved from digestion. Where did all that saved energy go?
Back to the brain. It created this amazing positive feedback loop. Fire leads to cooking. Cooking leads to softer food, smaller jaws, smaller guts, which saves energy. And then energy becomes available to fuel further brain growth. Fire literally helped fuel the expansion of the human brain.
Wow. Okay, so we've got free hands from bipedalism, fire changing our diet, an energy budget, a bigger brain. Did the free hands lead to other physical changes, maybe less obvious ones?
It did. Think about carrying things. Once our hands were free, thanks to opposable thumbs, we didn't need our mouths for carrying anymore. Carnivores, for example, often have large canine teeth, partly to grip and carry prey.
Right, you see wolves carrying chunks of meat.
Exactly. And when they close their mouths, those big canines often fit into a little gap in the opposite jaw. That gap's called a diastema. But once humans started carrying things with their hands, food, tools, babies, those large pointy canines weren't as critical. They gradually got smaller, and that diastema, it disappeared in our lineage.
It's just amazing how one change cascades into others. It really feels like our biology shaped our culture, like tool use, and then culture, like fire and cooking, turned around and reshaped our biology.
That's precisely the core idea behind the biocultural approach in physical anthropology.
Mm.
It doesn't see biology and culture as separate boxes. It sees them as two sides of the same coin, constantly interacting, constantly influencing each other. You really can't understand human evolution or even humans today without looking at both.
So it's like a bridge between the biological sciences and the social sciences.
Exactly. It forces you to consider both. You can't just look at genes, and you can't just look at behavior or tools. It's the interplay.
Can you give us some more examples of how looking through this biocultural lens helps?
Sure. We've already touched on human evolution. Brain growth, biology, enabled tool making, and fire culture, which led to smaller jaw jets biology, freeing up energy for more brain growth biology, enabling more complex culture, like language. It's a constant loop.
Okay, evolution makes sense. What else?
Human variation. Why do people look different around the world? It's partly genes, sure, biological inheritance, but it's also lifestyle, culture, diet, activity levels, exposure to sun affecting skin color over generations. It's both.
Right, nature and nurture interacting.
Precisely. Then there's human adaptation. How do humans live everywhere, from deserts to the Arctic? Let's take Tibetan highlanders living at extreme altitudes. Biologically, they've adapted. Many produce more red blood cells to capture the scarce oxygen.
Okay, that's the biology.
But culturally, they also adapt. Their diet is often rich in iron, which helps hemoglobin production.
Hmm.
And traditionally, pregnant women might move to lower valleys during pregnancy to protect the fetus from hypoxia. That's a cultural practice layered onto a biological adaptation.
So it's biology and behavior working together for survival.
Absolutely. Fourth area, disease and health. Why do certain diseases hit some populations harder? Think about cardiovascular issues in India, even affecting younger people. There's evidence for a genetic variant, a biological factor common in the population that increases vulnerability.
Okay, genetic predisposition.
Right. But it's often triggered or worsened by cultural factors, changing diets towards more processed foods, more sedentary lifestyles, maybe even environmental factors like air quality, which are linked to human activity. It's the combination. Similar patterns exist for things like diabetes risk or even TB susceptibility in certain tribal groups facing displacement stress.
It's a powerful and sometimes sobering connection and reproduction.
Same interplay, reproduction and fertility. Biologically, factors like a woman's uterine health influence her capacity to have children. But culturally, societal norms play a huge role. Some cultures are strongly pronatal, encouraging large families' children seen as assets or blessings.
While others might be more antinatal.
Exactly. Especially in some urban settings where the cost of raising children is high, they might be seen more as liabilities, leading people to choose smaller families. Biology provides the potential, culture shapes the outcome.
This interconnectedness is just fascinating. It makes me wonder about the field itself. How did this study evolve? What's the story behind the term physical anthropology? Did it always have this biocultural focus?
Not initially, no. The terminology actually reflects the field's evolution. The original term physical anthropology really took shape after the Enlightenment, especially as Europeans encountered more fossils and diverse human groups through exploration and colonialism. The focus then was very much on, well, the physical. Morphology, the form and structure of skeletons, bodies, and morphometrics taking measurements like skull size, height, limb proportions.
So classifying people based on what you could see and measure.
Pretty much. Comparing fossils, comparing living populations, trying to figure out evolutionary relationships and racial classifications based on physical traits.
But that sounds quite limited compared to what you're describing now.
It was. And that led to a big shift in the 1950s, spearheaded by a guy named S.L. Washburn. He argued for what he called the new physical anthropology. His point was we can't just look at bones and calipers. We need a more holistic interdisciplinary approach.
Bringing in other sciences.
Exactly. Integrating genetics, which was exploding then. Chemistry, physics, think carbon dating for aging fossils. Geology, stratigraphy, helping understand the layers where fossils are found. Paleopathology, studying diseases in ancient remains. Even medical sciences.
So broadening the toolkit massively.
Massively. And crucially, Washburn really pushed the idea that humans are both biological and cultural beings, that you have to study that interaction. That biocultural perspective became central to this new approach.
So is new physical anthropology the term we use now?
Well, no, because by the 1990s, what Washburn called new wasn't new anymore. It was just how the field was. So the consensus term shifted again to biological anthropology. It implies that integration of genetics, evolution, biocultural perspectives, it's all just part of the standard toolkit now. It reflects the mature integrated science it has become.
Okay, biological anthropology it is. Let's zoom right in now, down to the absolute basics, cells and the genetic material inside them.
Right, down to the nitty-gritty. The fundamental unit is the cell. Evolutionarily, the earliest were prokaryotes, think bacteria, amoeba. They showed up maybe 3.7 billion years ago. Simple, no defined nucleus.
Ancient history.
Truly. Then, much later, maybe 1.2 billion years ago, eukaryotes evolved. These have a proper nucleus containing the genetic material. All human cells are eukaryotes.
And within us, there are different types of eukaryotic cells.
Two main categories for genetics. Somatic cells are basically all your body cells, skin, muscle, nerve, everything except sperm and egg. They're diploid, meaning they have 23 pairs of chromosomes, 46 total, one set of 23 from your mom, one set from your dad.
Okay, pairs in somatic cells.
And then you have the germ cells, or sex cells, sperm, and eggs. These are haploid. They only have 23 individual chromosomes, not pairs.
Ah, so when sperm meets egg.
The 23 from sperm combine with the 23 from egg to make the first diploid cell. the zygote, with its full 23 pairs. And that zygote then divides and divides to create all the somatic cells.
Got it. And inside these cells, what are the key parts for understanding our biology and evolution?
For us, a few organelles are key. First, mitochondria. Everyone knows them as the powerhouse making energy. But anthropologically, they're super important because they contain their own separate DNA, mitochondrial DNA or mDNA.
Separate from the DNA in the nucleus.
Yep. And here's the crucial bit. mDNA is inherited almost exclusively from the mother. The sperm's mitochondria are usually in the tail, which gets discarded during fertilization.
So mDNA traces the maternal line.
Exactly. It's passed down mother to child, mother to child. Plus, it mutates at a relatively known, steady rate, and it's quite stable. This led scientists compare mDNA across populations and trace lineages back in time.
Is that where the mitochondrial Eve idea comes from?
Precisely. By comparing mDNA variation worldwide, researchers propose that all living humans descend matrilineally from a woman, or a small group of women, who lived in Africa roughly 200,000 years ago. It's like a molecular clock for our maternal ancestry.
Incredible. Okay, mitochondria and mDNA. What else?
We need to know about ribosomes. These are tiny structures, the sites where proteins are actually built. Think of them as the cell's protein factories. And then the main event, the nucleus. That's the cell's control center. It holds the vast majority of our genetic material, DNA and RNA. This is where our identity, our evolutionary history, is all coded.
DNA and RNA. The big ones, what are they, chemically speaking?
They're long-chain organic molecules. DNA, deoxyribonucleic acid, is famous for its double helix structure like a twisted ladder, two strands wound around each other. RNA, ribonucleic acid, is typically single-stranded.
And they're built from smaller units.
Yes, both are made of repeating units called nucleotides. Each nucleotide has three parts: a sugar molecule, a phosphate group, and a nitrogenous base. Those bases are the letters of the genetic code.
A, T, G, C, and U.
Right. There are two types of bases: purines, adenine A and guanine G, and pyrimidines, cytosine C, thymine T, and uracil U.
Okay, so 5 letters in total.
5 letters, but they pair up specifically. In DNA, adenine A always pairs with thymine T. They form a bond. Guanine G always pairs with cytosine C. They form a slightly stronger bond. This specific pairing is key to DNA structure and replication.
A with T, G with C. Got it. What about RNA?
In RNA, thymine T is replaced by uracil U. So in RNA, adenine A pairs with uracil U. Guanine G still pairs with cytosine C. Because the other strand can always be figured out from one strand due to these strict pairing rules, we often just write out the sequence of one DNA strand to represent the information.
Right. Knowing one side tells you the other. Now, the story of discovering this structure, the double helix, it's not just a straightforward science tale, is it? There's some human drama involved.
Oh, absolutely. It's a really compelling story, and it does say a lot about how science works and sometimes how credit gets assigned. A key figure was Rosalind Franklin. She was a brilliant chemist using X-ray diffraction to study DNA's structure.
X-ray pictures of DNA.
Essentially, yes. Her photograph 51 was incredibly clear and provided crucial evidence for the helical structure. It showed the repeating pattern.
So she had the key data.
She had incredibly important data.
Yeah.
But her colleague, Maurice Wilkins, showed photograph 51 seemingly without her full knowledge or permission to James Watson and Francis Crick. That photo, combined with other chemical knowledge, helped them build their famous double helix model.
And Watson, Crick, and Wilkins got the Nobel Prize in 1962.
They did. Rosalind Franklin tragically died of cancer in 1958, and Nobel Prizes aren't awarded posthumously. But even beyond that, her critical contribution was significantly underplayed for decades.
It sounds like a classic case of someone not getting the credit they deserved.
It really does. It's
Yeah.
often cited as an example of the Matthews effect, in science, basically. The idea that established figures or those with more institutional privilege, in this case perhaps male scientists at prestigious universities, tend to get disproportionately more credit, while others, perhaps women or those less established, might lose out, even with crucial contributions. It's a reminder that science is a human endeavor with all the complexities that entails.
A really important point. Okay, so we have this DNA structure. Now, how does life actually use this information? You mentioned the central dogma of genetics.
Right. The central dogma is the fundamental flowchart. DNA makes RNA, and RNA makes protein. That's the core principle. It happens through 3 main processes we need to understand.
Okay, process number 1.
DNA replication. This is how DNA makes a copy of itself.
And why does it need to do that?
For growth and repair. Remember, we start as one cell, a zygote. To become a 37 trillion cell adult, that cell has to divide and divide and divide. Before each division, the cell has to copy its entire DNA library perfectly, so both new cells get a complete set of instructions.
Makes sense. How does it actually copy itself? Like a photocopier?
Sort of, but chemically. It happens in three main steps. First, initiation. An enzyme called helicase acts like a zipper, unwinding and separating the two DNA strands by breaking the bonds between the bases. Other proteins jump in to keep the strands from zipping back up. Then another enzyme lays down a short RNA primer, like a starting block, creating a Y-shaped structure called the replication fork.
Okay, unzipped and primed. Step 2.
Elongation. This is where the main copying enzyme DNA polymerase gets to work. It reads the original strand and builds a new complementary strand alongside it, following those ATGC pairing rules. It builds the new strand in a specific direction.
Is it smooth sailing on both strands?
Not quite. One strand, the leading strand, gets copied continuously as the DNA polymerase follows the unzipping fork. But the other strand, the lagging strand, is tricky because it has to be copied in the opposite direction, sort of backwards, away from the fork. So it gets copied in short little chunks called Okazaki fragments.
Okay, leading strand smooth, lagging strand choppy. Then what?
Termination. Another enzyme removes all those RNA primers. DNA polymerase fills the gaps with DNA bases. And finally, an enzyme called DNA ligase acts like molecular glue, sealing up all the gaps, especially patching together those Okazaki fragments on the lagging strand to create two continuous, complete DNA molecules.
And each new DNA molecule has one old strand and one new one.
Exactly. That's why replication is called semi-conservative, and each new double helix conserves half of the original molecule.
It sounds incredibly efficient, almost perfect. But you mentioned earlier this process is somehow linked to aging and death.
Ah, yes. This is where that lagging strand comes back to bite us, so to speak. Because of the way it's copied in fragments and the need for primers at the start of each fragment, the very end of the lagging strand can't always be fully copied. In each round of replication, the lagging strand tends to get just a tiny bit shorter.
One base pair shorter, maybe? Seems tiny.
It is tiny, one base out of billions. But think about it. This happens every time a cell divides. From zygote to 37 trillion cells, that's a lot of divisions. Over a lifetime, this shortening accumulates at the ends of our chromosomes, in regions called telomeres. Hopefully, this shortening can start to eat into actual genes near the ends.
So essential genetic information could be lost over time.
Potentially, yes, or damaged. Genes responsible for vital functions, making insulin, coordinating immune responses could be affected. Add to that, as we age, the enzymes responsible for proofreading the replication process, catching errors, they also become less efficient. They wear down.
So chromosomes get shorter and more mistakes creep in.
Exactly. It's a double whammy. This combination of telomere shortening and reduced error correction means our cells gradually lose functionality. Their ability to divide properly decreases. This contributes to aging, increased risk of genetic disorders, and ultimately makes death an inherent, unavoidable biological process. We're basically programmed with an expiration date at the molecular level. Anti-aging tech might slow it, but the fundamental process is built in.
Wow. So, okay, DNA replicates itself with this built-in aging mechanism, but how do those sequences of ATGC actually do anything? How do they become the proteins that build us and run us?
Right, that's the next stage. Protein synthesis. And it involves the other two key processes, transcription and translation. First, let's quickly define proteins. They're the real workhorses. There are long chains of building blocks called amino acids. There are 20 standard amino acids used in our bodies.
And we need to get some of those from food, right?
Yep. 11 are non-essential. Our bodies can make them, but 9 are essential. We absolutely must get them from our diet. Things like histidine, leucine, lysine.
Okay, 20 amino acids making up proteins. What kinds of proteins are there?
Broadly, two types relevant here. Structural proteins literally build things. Your hair, your bones. They even determine your blood type antigens, like ABO. They provide the physical structure.
And the other type?
Regulatory proteins, these are crucial. They control the activity of the structural proteins and other cellular processes. They ensure things happen in the right way at the right time.
Like what? Give me an example.
Okay, think about growth. Regulatory proteins tell your body when to stop growing taller. They trigger puberty at adolescence. During fetal development, they're the ones telling initially identical stem cells. Okay, you become a heart cell, you become a lung cell, you become a brain cell. They orchestrate the whole developmental pattern.
They're like the project managers of the body.
That's a great way to put it. Now remember the gene. We said DNA is like a huge industrial area. A gene is like one specific factory in that area. It's a functional segment of DNA that contains the instructions for building one specific protein. You have an insulin gene factory that makes insulin protein, a hemoglobin gene factory making hemoglobin, and so on.
So when the body needs a specific protein, say more insulin after a meal, how does it get the instructions from the insulin gene factory?
That's transcription. Step one of protein synthesis. It's like the factory manager, the gene, writing down the instructions. An enzyme called RNA polymerase binds to the start of the gene. This signals the DNA double helix for that gene segment to temporarily unwind and separate.
Okay, the gene opens up.
Then, RNA polymerase moves along one strand of the DNA, reading the sequence of bases, A, T, G, C. As it reads, it builds a complementary strand of messenger RNA, mRNA, using those RNA pairing rules. A pairs with U, G pairs with C. This mRNA molecule is essentially a single-stranded copy of the gene's instructions.
So mRNA is the message.
It's the message. Once the RNA polymerase reaches the end of the gene, a stop sequence, the newly made mRNA molecule detaches from the DNA. The DNA zips back up, and the mRNA message is ready to leave the nucleus.
But wait, you mentioned something earlier. This mRNA isn't quite ready yet. Something about exons and introns.
Ah, yes. Good catch. The mRNA that's first transcribed, called pre-mRNA, often contains segments that don't actually code for the final protein. These non-coding segments are called introns. The parts that do code for the protein are called exons.
So the introns need to be removed.
Exactly. There's a processing step where the introns are spliced out and the exons are joined together to create the final, mature mRNA molecule that actually carries the protein recipe.
Okay, that makes sense. But didn't you also say this splicing helps explain how we get so many proteins from relatively few genes? Like 20,000 genes making 50,000 plus proteins?
Yes. This is the really clever part. Alternative splicing. It turns out that for many genes, the exons can be spliced together in different combinations. From the same pre-mRNA molecule, the cell can sometimes create several different versions of mature mRNA by including or excluding certain exons or joining them in a different order.
So one gene can actually produce instructions for multiple different but related proteins.
Precisely. It's incredibly efficient. It's like having one blueprint that you can use to build several slightly different models. That's how we get such protein diversity from a limited number of genes.
Nature is pretty smart. Okay, so we have this processed mRNA message carrying the recipe for a specific protein. It leaves the nucleus. Now what? How does it get turned into an actual protein?
That's the final step, translation. This happens out in the cytoplasm on those ribosomes we mentioned, the protein factories.
The chef gets the recipe.
Exactly. The ribosome is the chef, the mRNA is the recipe card, and we need delivery people to bring the ingredients. Those are transfer RNA, tRNA molecules.
Transfer RNA. What do they transfer?
Amino acids. Each tRNA molecule is designed to carry one specific type of amino acid. And crucially, each tRNA also has a 3-base sequence called an anticodon that matches a specific 3-base codon on the mRNA recipe.
Okay, codons are the words in the mRNA recipe, 3 letters long.
Perfect analogy. The ribosome reads the mRNA recipe 3 bases at a time. Each 3-base codon specifies which amino acid should be added next to the growing protein chain. Remember, there are 64 possible codons, 4 bases taken 3 at a time.
But only 20 amino acids. So some amino acids must have more than one codon.
Yes, most do. There's redundancy built into the code. Several different codons can signal for the same amino acid. There are also specific start and stop codons.
Start and stop, like punctuation.
Exactly. Translation begins when the ribosome finds a specific start codon, usually AUG, on the mRNA. This codon also brings in the first amino acid, methionine. The ribosome then moves along the mRNA, reading the next codon.
And a tRNA brings the right amino acid.
Yep. The tRNA with the matching anticodon docks onto the ribosome, delivering its amino acid. The ribosome then links this new amino acid to the previous one, forming the beginning of the protein chain.
And it just keeps going, codon by codon.
It chugs along. The ribosome shifts, reads the next codon, the correct tRNA arrives, the amino acid is added, the chain gets longer, peptide bonds form between the amino acids. This is elongation.
Until when?
Until the ribosome hits one of 3 specific stop codons, UAA, UAG, or UGA. There are no tRNAs that match these codons. They're like a full stop at the end of the recipe.
So ribosome just stops.
It signals the end. The ribosome releases the completed protein chain, the polypeptide, and detaches from the mRNA. The protein then folds up into its specific 3D shape to become functional.
And that's how a gene becomes a protein. DNA to RNA to protein.
That's the central dogma in action. Though sometimes things go wrong. Remember aging and mutations. Sometimes a mutation can accidentally create a stop codon in the middle of the mRNA sequence.
Oh, so the protein would be cut short?
Exactly. Translation stops prematurely, leading to an incomplete, likely non-functional protein. This is one way mutations can cause problems, especially as we get older and accumulate more errors.
Wow. We have covered an incredible amount of ground today. From, realizing we're 98% chimp, genetically speaking, to the tiny molecular machines copying DNA and building proteins inside our cells.
It's quite a journey, isn't it? And seeing how inseparable our biology and culture really are, how things like fire or social structure are written into our evolutionary history, right alongside changes in our genes or anatomy.
Absolutely. And connecting it all, seeing how something as fundamental as DNA replication with that little bit of shortening each time holds the key to our own mortality, it's profound.
It really is. And understanding how those subtle genetic variations interacting with our environment and our cultural choices,
Yeah.
diet, lifestyle influence everything from how we look to what diseases we might be susceptible to.
So, what's the takeaway for you listening? Well, maybe next time you look in the mirror, take a second to think about the millions of years of evolution packed into you, the incredible biological trade-offs, the ingenious solutions, the deep impact of culture shaping the physical being that is you.
And maybe this final thought to chew on. Given what we now understand about this inherent programmed aging built into our DNA replication, this sort of unavoidable cellular decline, what does that mean for our modern relentless pursuit of radically extended lifespans through technology? Are we trying to overcome something fundamental in our evolutionary design? Can we truly defeat aging, or are we just trying to postpone an inevitability written into our very cells?
Something to think about. We really hope this deep dive has given you a new appreciation for just how complex, elegant, and frankly amazing human life is, and maybe spark some curiosity to learn even more. Thanks for joining us. Until next time.